Hello everyone, my name is Hadi Zafar and welcome to First Aid Explained, where I, as a medical student, try my best to explain every page of the First Aid USMLE Step 1 2021 textbook. In this video, I'll be covering the biochemistry portion, pages 72 to 78. So let's begin. So here on the first page, we have something called ethanol metabolism. Now, ethanol is mainly oxidized to acetaldehyde through three different enzymes: alcohol dehydrogenase, cytochrome P450-2E1, and catalase. Catalase being in the peroxisome, alcohol dehydrogenase being in the cytosol, and P450-2E1 in the microsome. Now, to avoid acetaldehyde's toxic effects, after it is produced, acetaldehyde must be oxidized again to acetate by aldehyde dehydrogenase, and this occurs in the mitochondria. Now, keep in mind that females are more susceptible than males to effects of alcohol due to decreased activity of gastric alcohol dehydrogenase, decreased body size, and decreased percentage of water and body weight. Now, ethanol metabolism mainly occurs in the liver, where there's an increased NADH and NAD plus ratio, and this inhibits the TCA cycle, leading to high levels of acetyl coenzyme used in ketogenesis. So you lead up to ketoacidosis and high levels of alcohol, and lipogenesis, hepatosteatosis. Now, when ethanol overdose or alcohol abuse occurs, there's an increased NADH and NADH ratio in the liver, causing lactic acidosis, which is increased pyruvate conversion to lactate, fasting hypoglycemia because of decreased gluconeogenesis due to increased conversion of OAA to malate, ketoacidosis, and hepatosteatosis. And here you can see in the diagram glucogenesis and glycolysis pathways, as well as the acetyl coenzyme A entering the TCA cycle. And the arrows in red are pathways stimulated by increased NADH and NAD ratio, and the dashed lines are pathways inhibited by increased NADH and NAD ratio. Now, fomepizole blocks alcohol dehydrogenase and is the preferred antidote for overdoses of methanol or ethanol glycol. Now, alcohol dehydrogenase has higher affinity for ethanol than for methanol or ethanol glycol. Ethanol can be used as a competitive inhibitor for alcohol dehydrogenase to treat methanol or ethanol glycol poisoning. Now, disulfiram blocks acetaldehyde dehydrogenase, leading to increased acetaldehyde. This leads to increased hangover symptoms and discouraging drinking. So we give disulfiram for patients who are trying to reduce their alcohol intake. Alright, moving on. Now here we have the biochemistry metabolism, and these are the terminologies used. So first we'll talk about the enzyme terminology. Now, the enzyme name often describes its function. For example, glucokinase is an enzyme that catalyzes the phosphorylation of glucose using a molecule of ATP, so glucokinase. Now, kinase, just the kinase word, catalyzes transfer of a phosphate group from a high-energy molecule, usually ATP, to a substrate, for example, phosphofructokinase. Phosphorylase adds an organic phosphate onto substrate without using ATP. So glycogen phosphorylase adds phosphate without using ATP. Phosphatase removes phosphate group from substrate. Dehydrogenase catalyzes oxidation reduction reactions. Hydroxylase adds hydroxyl group OH onto the substrate, for example as tyrosine hydroxylase. Carboxylase transfers CO2 groups with the help of biotin. So, for example, of enzyme is pyruvate carboxylase. Now, mutase relocates a functional group within a molecule. For example, vitamin B12 dependent methylaminyl-CoA mutase. Synthase or synthetase joins two molecules together using a source of energy, usually ATP. Now we have the rate-determining enzymes of metabolic processes. So the process of glycolysis, the enzyme is phosphofructokinase 1, and this is the rate-determining enzyme in this process. And the regulators are AMP and ATP. Gluconeogenesis is fructose 1,6-biphosphatase 1, and the regulator is AMP. TCA cycle, the rate-determining enzyme is isocitrate dehydrogenase. Glycolysis is glycogen synthase. Glycogenolysis is glycogen phosphorylase. In the HMP shunt, glucose 6-phosphate dehydrogenase, or G6PD, and the regulators are NADP and NADPH. De novo pyrimidine synthase is carbamoyl phosphate synthase 2. De novo purine synthase is glutamine phosphoribosyl pyrophosphate, or PRPP. Urea cycle is carbamoyl phosphate synthase 1. Fatty acid synthesis is ACC, or acetyl-CoA carboxylase. Fatty acid oxidation, the regulator enzyme or redetermining enzyme, is carnitine acetyltransferase 1. Ketogenesis is the HMP HMG CoA synthase, and cholesterol synthase is HMG CoA reductase. Okay. Now we have the metabolism sites. So there's mitochondria, cytoplasm, or both. So in mitochondria, we have fatty acid oxidation or beta oxidation, acetyl-CoA production, TCA cycle, oxidative phosphorylation, and ketogenesis. Whereas in the cytoplasm, we have glycolysis, HMP shunt, the synthesis of cholesterol, and the smooth endoplasmic reticulum, proteins, ribosomes, and rough endoplasmic reticulum, fatty acids, and nucleotides. In both metabolism sites in mitochondria and cytoplasm, we have heme synthesis, urea cycle, and gluconeogenesis. The mnemonic for this is HUGS take 2, 2 for both. HUGS, heme synthesis, U for urea cycle, and G for gluconeogenesis. And here we have the summary of pathways from galactose metabolism, gluconeogenesis, glyconeogenesis or glycogenolysis, HMP shunt, fructose metabolism, lipid metabolism, ketogenesis, protein metabolism, TCA cycle, and the urea cycle. So this is the summary of all the pathways, which is important for your exam. All right, moving on. Now here we have how ATP is produced. Now, ATP production is the aerobic metabolism of one glucose molecule that produces 32 net ATP via malate aspartate shuttle. Now, 30 net ATP via glycerol 3-phosphate shuttle, which is the muscle. And anaerobic glycolysis produces only 2 net ATP per glucose molecule. So aerobic metabolism produces 32 for the heart and liver via the malate aspartate shuttle, and 30 net ATP via the muscle for the glycerol 3-phosphate shuttle. And anaerobic glycolysis produces only 2 net ATP per glucose molecule. Now, ATP hydrolysis can be coupled to endogenetically unfavored reactions. Now, arsenic, a poison, causes glycolysis to produce 0 net ATP. Now let's move on to the activated carriers. So we have the carrier molecule and the carried in the activated form. So the carrier molecule ATP, carried in activated form is phosphoryl groups. NADH, NADPH, and FADH2 carries electrons. CoA and lipoamide carry acyl groups. Biotin carries carbon dioxide. Tetrahydrofolates carry 1 carbon units. And SAM, or S-adenosine methanion, carries CH3 groups. And TPP carry aldehydes. Now, we have something called as the universal electron acceptors. Now, nicotinamides, NAD+, or NADP+, from vitamin B3, and flavonucleotides, or FAD, from vitamin B2. And NAD+ is generally used in catabolic processes to carry reducing equivalent away from NADPH. Now, NADPH is used mainly in anabolic processes, for example, in steroid and fatty acid synthesis, as a supply of reducing equivalents. Now, NADPH is a product of the HMP shunt. NADPH is used in the anabolic process, respiratory burst, cytochrome P450 systems, and glutathione reductase. Now, what is the difference between hexokinase versus glucokinase? Now, the phosphorylation of glucose to yield glucose 6-phosphate is catalyzed by glucokinase in the liver and hexokinase in other tissues. Hexokinase sequesters glucose in tissues, where it is used even when glucose concentrations are low. At high glucose concentrations, glucokinase helps to store glucose in the liver. Glucokinase deficiency is a cause of maturity, onset obesity, or onset of diabetes of the young, MODY, and gestational diabetes. So if there's a glucokinase deficiency, you have a likely onset of diabetes of the young. So hexokinase is mostly in the tissues and except in the liver. So it's in the tissues but not in the liver, nor it's in the pancreatic beta cells. It has a lower affinity, it has a lower Km, so a higher affinity. It has the Vmax is lower, so a lower capacity, and is not induced by insulin. And the feedback inhibition is glucose 6-phosphate. Now glucokinase, to help remember this, the gluco for the L in glucokinase helps remember that the location is in the liver and the beta cells of the pancreas. And has a higher Km, leading to a lower affinity, and a higher Vmax, leading to a higher capacity. And it is induced by insulin. And the feedback inhibition is by fructose 6-phosphate, compared to glucose 6-phosphate in hexokinase. Okay. Now we have glycolysis regulation in the key enzymes in glycolysis regulation. So the net glycolysis happens in the cytoplasm. And we have the following unbalanced equation. Now the equation is not balanced. And the exact balanced equation depends on the ionization state of the reactants and products. So the part in glycolysis regulation that require ATP is for the conversion of glucose to glucose 6P, right? Which is done by hexokinase. And fructose 6P turning to fructose 1,6-BP, which is done by phosphorofructokinase 1 and is the rate limiting step. Now, regulation that produces ATP is either the forward or backward production of 1,3-BPG or 3-PG with the enzyme of phosphoglycerate kinase, and phosphoenolpyruvate to pyruvate with the pyruvate kinase. Now the regulation by fructose 2,6-biphosphate and phosphofructokinase are the same. Bifunctional enzyme whose function is reserved by phosphorylation by protein kinase A. Now these both enzymes are essential for the fasting and fed state of our body. Now in the fasting state, there's high levels of glucagon, which leads to high levels of CAMP, which in turn leads to high protein kinase A, increasing the enzyme FBPase2, which is fructose biphosphatase 2, and decreasing PFK2, which is phosphofructokinase 2, leading to less glycolysis and more gluconeogenesis. Whereas in the fed state, there's an increased level of insulin, leading to a decreased CAMP, leading to decreased protein kinase A, leading to a decrease of FBPase2, and an increase of PFK2, which is phosphofructokinase 2. So there's more glycolysis and less gluconeogenesis. So to remember each enzyme, FBP, which is fructose biphosphatase, is Fabian the peasant, and it has to work hard when starving, so the fasting state. And PFK, Prince Frederick, so phosphofructokinase 2, works only when fed. So PFK is only activated in the fed state. Now we have the pyruvate dehydrogenase complex. And this is a mitochondrial enzyme complex linking glycolysis and the TSA cycle, differentially regulated in fed, or considered active, or fasting or inactive states. Now the reaction is listed below. And this contains 3 enzymes requiring 5 cofactors. The 5 cofactors are thiamine pyrophosphate or vitamin B1, lipoic acid, CoA or vitamin B5, pantothenic acid, FAD for B2, riboflavin, and NAD, B3, niacin. And this is activated by increased NAD to NADH ratio, or increased ADP and increased calcium. Now, arsenic, again a poison, inhibits lipoic acid. So arsenic poisoning can disrupt the pyruvate dehydrogenase complex, and the clinical findings are pigmentary skin changes and skin cancer, vomiting and having diarrhea, and QT prolongation, and so-called garlic breath. So just imagine a vampire with pale skin and running away from a QT or QT prolongation with garlic breath. Okay, moving on. Now we have the pyruvate dehydrogenase complex deficiency, and this causes a buildup of pyruvate that gets shunted to lactate via LDH and alanine, which via ALT, and this is X-linked. So the findings are in these patients with pyruvate dehydrogenase complex deficiency are neurological defects, lactic acidosis, and increased serum alanine starting in infancy. And the treatment for this is an increased intake of ketogenic nutrients, for example, high fat content or increased lysine and leucine. Next, we have the pyruvate metabolism. Now, pyruvate is an important chemical compound in biochemistry. It is the output of the anaerobic metabolism of glucose, known as glycolysis. And keep in mind that one molecule of glucose breaks down into two molecules of pyruvate. And with the help of ALT or LDH, pyruvate gets shunted either to alanine, into the Cahill cycle, or into lactate in the Cori cycle. And the functions of different pyruvate metabolic pathways are listed here. So the first one, being alanine aminotransferase of vitamin B6, and this is alanine carries amino groups to the liver from muscle. And then vitamin B7 or pyruvate carboxylase, oxaloacetate, can replenish TCA cycle or can be used in gluconeogenesis. Now pyruvate dehydrogenase, which is vitamin B1, B2, B3, B4, B5, lipoic acid, and this transition from the glycolysis to the TCA cycle. Lastly is lactic acid dehydrogenase, vitamin B3, and this is the end of the anaerobic glycolysis, and this is the major pathway in RBCs, WBCs, kidney, medulla, lens, testis, and cornea. Lastly, we have the TCA cycle, also known as the Krebs cycle, and this is where pyruvate eventually gets converted to acetyl-CoA, producing one NADPH and one carbon dioxide. Now the TCA cycle produces 3 NADH, 1 FADH2, 2 carbon dioxides, 1 GTP per acetyl-CoA, and 10 ATP per acetyl-CoA. Now TCA cycle reactions occur in the mitochondria, and alpha-ketoglutarate dehydrogenase complex requires the same cofactors as pyruvate dehydrogenase complex, so vitamins B1, 2, 3, and 5, as well as lipoic acid. A nice mnemonic is that citrate is Krebs' starting substrate for making oxalate. And this will help you remember all the essential substrates in the TCA cycle. Starting from citrate, and then citrate is, so I, isocitrate, then K for Krebs, so alpha ketoglutarate, and then Krebs starting, so S succinyl-CoA, and then substrate, so succinate, then 4 making, so 4 F for fumarate, M making for malate, and then oxyl acetate, so this is the final product. Okay. Now we have the electron transport chain and oxidative phosphorylation. Now, NADH electrons from glycolysis enter mitochondria via the malate, aspartate, or glycerol 3-phosphate shuttle. FADH2 electrons are transferred to complex 2 at a lower energy level than NADH. The passage of electrons results in the formation of proton gradient that, coupled to oxidative phosphorylation, drives the production of ATP. So you can see here that this whole electron transport chain happens in the mitochondria. So the mitochondrial matrix is outside, inner mitochondrial membrane, and the intermembrane space. Now the ATP produced via ATP synthase, so 1 NADPH equals to 2.5 ATP, whereas 1 FADH2 is equal to 1.5 ATP. So 2.5, 1.5. Now for the poisons that affect oxidative phosphorylation, so electron transport inhibitors, this leads to directly inhibiting electron transport, causing a decreased proton gradient and blocking of ATP synthesis. So rotenone, a complex 1 inhibitor, antimycin, a complex 3 inhibitor, and cyanide, carbon monoxide, and azide, the ides, 4 letters, inhibiting complex 4. Then we have ATP synthase inhibitors, and this directly inhibits mitochondrial ATP synthase, causing an increased proton gradient, but no ATP is produced because electron transportation stops. And oligomycin is the poison that causes ATP synthase inhibiting. Now we have uncoupling agents. This leads to increased permeability of membrane, causing a decreased proton gradient and increased O2 consumption. ATP synthesis stops, but electron transport continues, producing heat. So 2,4-dinitrophenol, which is illicitly used for weight loss, and aspirin, where the fevers often occur after overdose, and thermogenin in brown fat, which has more mitochondria than white fat. Now, let's talk about the irreversible enzymes in gluconeogenesis, which are pyruvate carboxylase, phosphoenolpyruvate carboxykinase, fructose 1,6-biphosphatase 1, and glucose 6-phosphate. So PPFG. So pathway produces fresh glucose. So for pyruvate carboxylase in the mitochondria, and this converts pyruvate to oxalate. And this requires biotin, ATP, and activated acetyl-CoA. Whereas phosphoenolpyruvate carboxykinase occurs in the cytosol, where oxaloacetate is converted to phosphoenolpyruvate, or PEP, and this requires GTP. Then we have fructose 1,6-biphosphatase 1. This is also in the cytosol, and this converts fructose 1,6-biphosphate into fructose 6-biphosphate. And then lastly, there's glucose 6-phosphatase, and this is in the endoplasmic reticulum, where this converts glucose 6-phosphate to glucose. Now, gluconeogenesis primarily occurs in the liver, and this serves to maintain glycemia during fasting, meaning steady levels of glucose. Now, enzymes are also found in the kidney, intestinal epithelium. Deficiency of the key gluconeogenic enzymes causes hypoglycemia, as the muscle cannot precipitate in gluconeogenesis because it lacks glucose 6-phosphatase. Now, odd-chained fatty acids yield 1-propionyl-CoA during metabolism, where it can enter the TCA cycle as succinyl-CoA and undergo gluconeogenesis, and serves as a glucose source. Now, to remember this, it's odd for fatty acids to make glucose. Even chained fatty acids cannot produce new glucose, since they yield only acetyl-CoA equivalents. Alright, I think that's it for today. Thank you all for listening, and see you guys in the next one. Thank you.